This is Unit 7.5, Hardy-Weinberg Equilibrium. In the last video segment, we talked about populations. Population is a group of individuals of the same species that live in the same area and can interbreed, producing fertile offspring. Now we're going to continue with this idea of populations. Population is the smallest unit that can evolve. In other words, individuals cannot evolve. Individuals are selected for, whereas populations evolve. Now before we get too far, we first need to address the concept of an allele. An allele is a version of a gene that is an inheritable unit that controls a particular feature of an organism. So for example, eye color. Now, you have received two alleles. We call that diploid. One you received from your biological father, one you received from your biological mother. It is the combination of these two alleles that result in the phenotype, or what we see. So you can see on the bottom of my screen here, I have a flower. The dominant allele is the purple, we represent by big W, and the recessive allele is the small w, which is the allele for white. As I said before, all organisms are diploid. They have two copies. One they received from their biological father, one they received from their biological mother. It's a combination of these that result in the characteristic that we see, such that if an individual is homozygous dominant, such that they have a big W and a big W, that will result in a purple flower. If they are homozygous recessive, small w, it will result in a white flower. If they are what we call heterozygote, a big W and a small w, they are also purple in color because the dominant allele, the big W, is able to mask the smaller w for white. So make sure we understand the difference between phenotype is what we see and genotype is what the actual genes are. We'll continue with the concept of an allele frequency. Allele frequency refers to how frequently a particular allele appears in the population. And that will be represented in the form of a decimal, such as a percentage. In order to calculate the allele frequency, we use the equation. The allele frequency of A is the total number of A alleles in the population divided by the total number of alleles present in the population. So we need to add both the dominant alleles, the big A, and the recessive alleles, the small a, and that will give us our frequency of that particular allele. As I said before, we have to be very careful when we're asked to find genotype, phenotype, and allele frequency, and that's really demonstrated well on this slide right here. So I have a population of 9 flowers, 7 are purple, 2 are white, and depending upon what the question is asking us, our results are going to be slightly different. So for instance, if the question asks what is the genotype frequency, our genotype frequency would be 6 of the 9 are homozygous dominant, big WW, or 67% or 0.67. 2 of the 9 are white, small w, or 0.22 or 22%. And then our heterozygotes, big W, small w, 1 of the 9 or 0.11 have that particular genotype. If the question asks what is the phenotype, well, there are 1, 2, 3, 4, 5, 6, 7 purples and 2 whites. 7 divided by 9 is 0.78. 2 divided by 9 is 0.22. That would be our frequency of purple or our frequency of white, respectively. And the last one is really the focus of today, the allele frequency. With the allele frequency, remember that all individuals contribute 2 alleles to the population. Similar to our genotype frequency, we just count the number of W's, the big W's, 13 of 18 for 0.72, and 5 of 18, which equals 0.28. Notice I represent these as a decimal. Now, if we go back to our earlier presentations, we were talking about evolution. And this is an example of microevolution. So basically evolution at the smallest scale. And in order for us to determine if the population that we're examining is in fact evolving, what we are going to observe is we're going to see a change in the allele frequency over generations. Let me show you how this works. On the left-hand side here, we see the original population. Once again, we have 7 purple flowers and 2 white flowers. The frequency of big W is 0.72. The frequency of the small w is 0.28. So this is the original population or the original generation. And we can examine this population many generations later. And once again, we look at the allele frequency. This time, however, what you'll notice is the big W's, there's only 8. So it went from 13 to 8. Whereas the small w's went from 5 to 10. This is an example of a population that is evolving. When we see a change in these allele frequencies, that would suggest our population is changing. Well, what happens if our allele frequency remains constant? If they remain constant, in other words, our population doesn't evolve, this is what we have a particular situation that is described as the Hardy-Weinberg Equilibrium. In order for Hardy-Weinberg Equilibrium to occur, these 5 conditions must be met. These include no mutations in the population, so that DNA doesn't change. No sexual selection. In other words, individuals will randomly mate. No natural selection. We have to have extremely large sized population. Otherwise, we're going to end up with a genetic drift, such as a bottleneck. And the final condition, no gene flow. No immigration or emigration. So in other words, we don't introduce or lose those unique traits from our population. If any one of these conditions is not met, microevolution occurs. Now, let's apply Hardy-Weinberg equilibrium. Now, I just want us to be aware that this is a hypothetical situation. I can't think of any scenario that this is actually true in nature. So in other words, there's really no non-evolving population. If this is true, remember that our allele frequencies must be the same. If I check the allele frequencies today, tomorrow, 10 years from now, that population is not going to change. Now, because this is hypothetical, it really is just a model, and we'll get into the concept of a null hypothesis later, but just remember that term. So why have an equilibrium if it never exists? Well, it really is just a tool that scientists use for actually measuring evolutionary change. So how does this work? This is just the overview from College Board. So let's apply the Hardy-Weinberg equilibrium. So we're going to look at cats. We have the black cat there, and we have the white cat. So let's assume we have 2 alleles. Big B represents the dominant trait for black, and small b represents the recessive trait for white. Now, we're going to assume that the dominant allele is represented by the letter P, and the recessive allele is represented by the letter Q. Dominant P, recessive Q. So if we add all the alleles in our population, the equation that we are going to use is P plus Q equals 1. This is the first equation that you'll need for solving Hardy-Weinberg questions. Now, College Board will give you this equation. I will give you this equation, so you don't have to remember it. remember it. But it's a very useful equation. P plus Q equals 1. So in other words, we add up all the alleles for that particular trait. The dominant plus recessive equals 1. Note that we can rearrange these equations. 1 minus P equals Q and 1 minus Q equals P. You may have to do that in a question. Our second equation is a little more complex. Now this occurs when we're actually counting individuals. Remember that our individuals can be homozygous dominant, big B in this instance. They can be homozygous recessive, small b, or they can be heterozygote, big B, small b. So if we put this together, remember big B, which is our dominant allele, is represented by P. P times P is equal to P squared. So the sum of our dominant homozygote dominant population is represented by P squared. For our homozygous recessive, remember, our recessive trait is represented by lowercase q. So Q times Q is equal to Q squared. Our white cats in our population would be represented by the mathematical equation Q squared. And lastly, our homozygotes. Well, remember that our homozygotes are represented in this instance by the dominant allele, big B, and the recessive allele, the small b. So P times Q. But remember that these can also be represented by small b, big B. And I'll tell you right now that the order of the alleles doesn't matter. So in order to justify this equation, we are going to simplify it, and the heterozygotes in our population are going to equal 2pq. So when we add all the frequencies of the individuals together, we come up with this equation here shown in the orange box. P squared, which is homozygous dominant, plus 2pq, which is our heterozygotes, plus q squared, which is our homozygous recessive, equals 1. And that is the second equation that you will need to use for Hardy-Weinberg equilibrium. Now, when we look at solving these equations, you'll probably have to use both sets of equations, and I'll give examples of how that works in later presentations. So, remember that the allele frequencies in the population can be calculated from the genotype frequencies. And once again, this is the equations that you'll receive from College Board on your formula sheet. As we continue, what I want you to remember is that changes in the allele frequency provide evidence for the occurrence of evolution of a population. So in this example right here, we can see our population of brown mice and black mice. Brown mice are represented by big A, which is the dominant trait, and black mice are represented by lowercase a, homozygous recessive. As you can see, in generation 1, we have an equal distribution of both the recessive trait, small a, and the dominant trait, big A. If we introduce a predator to our population, the fox, remember what I said, one of the characteristics of a population that is evolving is that we have natural selection. If we look at the second generation and the third generation, what you'll quickly notice is that fox seems to select for the black mice. And over generation 2 and finally in generation 3, what you'll notice is that we see a shift in the alleles in our population. Originally started off with 50% dominant A, 50% small a. We end up in the third generation with 0.9% big A and 0.1% small a. So we have demonstrated that the population is in fact evolving because natural selection is driving this evolution. Now, as we talked about in the population genetics presentation, I said that small populations are especially susceptible to random environmental impacts, more so than the larger population. And that is all because of genetic drift. So we look at our population here on the left-hand side. We have 0.3% the dominant allele and 0.7% the recessive, lowercase a. By random chance, I select 3 of the beetles, and by chance, I select 3 gray beetles. They are then going to create the next generation or the new population. And what you'll notice is because of random chance, because this is a small population, I have now eliminated the dominant A from the population. And now the population is 100% or 1.0 recessive allele. Once again, this does not happen in a larger population. This is a unique characteristic of a small population in genetic drift.